Now we will see the basic features of genetic code. We know that genetic code is a nucleotide sequence within the mRNA molecule. So the first feature of genetic code is that it's a triplet nucleotide in the mRNA. It's a triplet sequence. All right. So this concept was proposed by George Gamow, and he reached into this conclusion by his calculations. It's like we know that we have 20 amino acids in total, which has to be incorporated during translation to a protein. So what is the possibility that the 20 amino acids are encoded by the 4 nucleotide bases or 4 nucleotides? So the combination or the conclusion is that if a single base codes for an amino acid, then what is the possible amino acid, or what is the possible total amino acids in the protein? It is like 4:1. That means one base codes for one amino acid. If it is so, we will have only 4 amino acid. So that is not a standing statement. So that it's not a single base coding for an amino acid. All right. Then the second possibility is combination of 2 nucleotides. So if 2 nucleotides together codes for an amino acid, let's see what is the possibility. What is the total number of codons available? Will be 16. Right. So 16 codons will be there, and so 16 amino acids are possible in total. That also not sufficient to satisfy the total 20 amino acid. So then move to the third possibility, that it's a triplet. That means 3 nucleotides will code for a particular amino acid, or 3 nucleotides will act as a codon. And that will give you a total of 64 codons. Right. So this number actually essentially covers the number 20, which is the total number of amino acids. So that concept or that theory was accepted. Right. So we have 20 amino acids, and these has to be encoded by the 64 different codons. Okay. And we will move into the further discussions why it is 64 and we have only 20 amino acids, but there are 64 codons. How that will be correlated, that we will discuss in the next features. Okay. So this was cracked. This triplet codons has been cracked. That means which sequence, which combination codes for which amino acid. This has been cracked by the scientists. That means Heinrich Matrai and Marshall Nirenberg. And the result is shown in this table. Right. That means UUU codes for phenylalanine, CAU codes for histidine, like that. GGA codes for glycine. So in that fashion, the result is available now. Right. And actually this was evidenced, and this was prepared by an elaborate experiment, right, to crack the genetic code. And in that experiment, they found that the 20 amino acids has been encoded successfully by a specific codon. Right. And moreover, after the 20 number, the remaining codons will also code for certain amino acids which are already encoded by some other codons. That means certain amino acids are encoded by more than one codons, and in that way the number we will consider the number. Okay. It will be quite interesting to understand how they found that each codon codes for that particular amino acid. For that, actually they used a controlled translation experiment. in this controlled translation experiment, they provided all the required materials for translation to a translating system. That means it could be an organism, but they used different test tubes, at least 20 test tubes. So we have 20 test tube, right? So in each test tube, it's a translating system. That means we have all the machineries required for translation, and each test tube will be having a particular. And in the first experiment, in the beginning, they used poly U, as the poly U was the mRNA molecule used for translation in all the test tubes, right? And each test tube was different in the amino acid provided, right? It is like in the first test tube they provided, for example, alanine, and in here it is glycine, and here it may be phenylalanine, and here it may be methionine. So in that way they prepared an experimental setup. Okay, so you have 20 such test tubes, and they allowed them to carry out translation. And after translation, they found that this poly-U mRNA is actually providing or acting as a substrate, acting as a template for translation of an mRNA or a protein chain which contains only phenylalanine. That means there was a protein synthesis only in this particular test tube, right? And that test tube consists of a protein which is having polyphenylalanine, right? That's having a polyphenylalanine protein. That means the poly-U codes for polyphenylalanine chain, right? By that they reached into the conclusion that UUU sequence or a poly-U sequence codes only for phenylalanine, right? And already George Gamow has stated that it's a triplet codon. So it is easy to conclude that UUU sequence codes for phenylalanine, right? In this fashion, they cracked all the nucleotides. They used all the nucleotides as a single nucleotide sequence, and also in combination of 2 and combination of 3. And finally, they concluded into this fashion, or they could prepare a genetic code in this particular as shown in this particular table, right? So out of the 64 codons, 61 codons codes for amino acids. That means of the 64, the 61 codons are meaningful, right? And of the 61, one codon will act as the start codon, right? That is what the AUG, which actually codes for methionine, and that is the start codon. And the remaining 3, right? The remaining 3 codons actually does not code for any amino acid, and they are considered as the stop codons, right? We have the stop codons are UGA, UGG, and UAG, UAA, and UGA as the stop codons. All right. Now, the second feature is degeneracy of the genetic code. Okay, degeneracy of the genetic code. This will give you the answer for the question why there are 61 codons for the 20 amino acids. Okay, that means as per the first property, that means it's a triplet codon and there are 64 codons to encode the 20 standard amino acid. So what is the possibility of for the remaining 44 codons or the remaining codons? Actually, they code, that means certain amino acids are encoded by more than one codons. That's why we have more codons than the number of amino acids, right? So certain amino acids are encoded by more than one codon. This is what we denote by the degeneracy of genetic code. certain amino acids are encoded by more than one codon. Okay, then the third statement is about unambiguity. The genetic code is unambiguous. That means as a particular genetic code will code for a specific amino acid. It is highly specific. A single codon will code for only one amino acid, one and only one amino acid. It is highly specific, right? So that codon will never code for a second amino acid, right? You must not get confused. Degeneracy states that an amino acid will be coded by more than one codon, but specificity states that one codon will be specific for one amino acid. All right, so this will give you an idea. The degeneracy means that, you know, this amino acid 1, here the amino acid 1 is encoded by the codon 1, codon 2 and codon 3. Both these three codons codes for the amino acid 1. There is an actual example. GGU codes for glycine, GGC also codes for glycine, and GGA also codes for glycine. That means glycine is encoded by 3 codons. That's what degeneracy. The glycine is encoded by 3 codons. Okay. At the same time, what is specificity? Specificity states that GGU codes for glycine. Alright, and GGU never codes for any other amino acid. GGU codes only for glycine. And GGU cannot code for alanine or phenylalanine or any other amino acid. Similar is true for GGC. GGC codes for glycine alone. It will never code for any other amino acid. Right. So that is what the specificity. Clear. So hope the degeneracy and specificity is clear. And there is a concept called the wobble hypothesis that we will discuss in some other video. So actually the degeneracy of the genetic code is an attribute of the wobble position. Okay, actually why certain amino acids are encoded by more than one codon is a feature of the wobble position. And what is wobble position? Wobble position is actually the first base of the anticodon in the tRNA and the third base of the codon in the mRNA. That position is what we call wobble position. Okay, we will have a detailed discussion in the some other videos. Okay, so if the first codon, that means if the wobble position in the tRNA is uracil, then it can recognize adenine and guanine at the third position of the codon. Right, that means you can see that the interaction between the nucleotides at that position, that means the interaction between the third base of the codon and the first base of the anticodon is not that highly specific. It's not that strong as the interaction between the other two. Right, and this will give you some flexibility. There are some flexibility in interaction. That's what it says. Okay, so this image also will give you an idea, right? And in that way we have different codons for different amino acids. That's also shown in the table, right? Methionine is an amino acid with only single codon. Similarly tryptophan also, they have only single codons. You can refer back the table which are the codons, right? At the same time there are number of amino acids which have 2 codons, and there are certain amino acids like isoleucine with 3 codons, and a set of amino acids with 4 codons, and we have 3 amino acids with 6 codons. That is the maximum number of codons for an amino acid. Okay, and this will explain the wobble position and the degeneracy in detail, right? See, if the first position of the tRNA, the first position of the tRNA, that means the wobble position, if it is C, then that is highly specific. There is no degeneracy. If it is C, then it codes for only, or it will recognize only G at the third position of mRNA. If it is A, then also it codes or recognize only U in the mRNA. But if it is G at the first position of the tRNA, then it can recognize C and U. Right, it can recognize both C and U at the third position of mRNA. Similarly, if it is U, it can recognize A and G. And if it is I is actually a modified base. It's a modified nucleotide base. It's actually called inosinate. Right. So this can recognize three codons. That means C, A, and U will be recognized at the third position of mRNA. All right. So that's what gives you the explanation for degeneracy of genetic code. The fourth feature of genetic code is that it's a non-overlapping sequence. There is no overlapping. That means the explanation is given there. If AGC codes for an amino acid, then the next amino acid is coded by AUC. That means the next three letter will code for the next amino acid. And it is like AGC codes for an amino acid, then GCA codes for the second amino acid. It is not true. Right? It is not overlapping. That means once a triplet is coding for an amino acid, and the sequences or the nucleotides within that triplet will not be used for decoding of any other amino acids. Okay, that is the fourth statement. Then the fifth feature is there is no punctuation mark or there is no space between codons. There is no full stop, there is no comma, there is no other marking, or there is no space usually between codons. Okay, it's a continuous sequence within the mRNA molecule. And finally, this one is universal. Genetic code is actually universal. In fact, it is now we consider them as nearly universal, not completely universal, but it's nearly universal. What does it mean? Universality states that if a codon is coding for example, if a codon codes for amino acid X, right, and this will be true in all the living forms, like in bacteria, in eukaryotes, plants, animals, and irrespective of the location. Right? In a particular region, like in Africa, it codes for the amino acid X. In Asia, also it will be coding for the same amino acid, and in America, also it will be coding for the same amino acid. And at any place, it will be having the same information. That's what the universality. Irrespective of the location and irrespective of the organism, the sequence or the codon will be coding for the same amino acid. Okay. Even though we say that it is universal, we can find certain variations of this universality. Right, these are some examples given there. Like UGA actually code it's a stop codon. Right, UGA actually is a stop codon, but in certain organisms like mycoplasma and also in mitochondria of certain species, this UGA codes for tryptophan. Right, so that's a deviation. So it's a deviation from the universality of genetic code. Similarly, another stop codon UGA sometimes codes for cysteine in certain organisms. So similarly for leucine, the codon for leucine like CUG codes for threonine in mitochondria of yeast. So these are certain variations from the universality. Okay, otherwise it's having a universal capacity. Right. So and also one added point is that there are two amino acids, two special amino acids considered like selenocysteine and pyrrolysine. These are amino acid in addition to the 20 amino acids usually found in the proteins. Right, in proteins of a biological system, we can see two more amino acids. These are selenocysteine and pyrrolysine. Right, and actually this selenocysteine is sometimes encoded by the stop codon UGA. Okay, actually it's a stop codon and it has to be terminated at that point. But in certain organisms, it codes for a modified base called or modified amino acid called selenocysteine. Okay, and because of this property, the selenocysteine is considered as a 21st amino acid by certain authors. All right, so by this we'll come to the end of this part of presentation of the features of genetic codes. So I'll recall that genetic code is a triplet codon. It is degenerate. It is unambiguous. Then there is no space or punctuation between them, right? And it is having a universal feature. All these are the different features of the genetic code. Okay, fine. Thank you for listening.